Here I will show you TEtrimmer. I'm Jiang Zhao from FT Hainan University. First of all, I want to say many thanks to all the guys who contributed to this tool, especially the authors on the paper. So this is not a tutorial video. I'm not to show you how to run this step by step, but give you a general introduction. I will provide another video to show you how to understand outputs and how to run TEtrimmer GUI for the further curation. Transposable elements are DNA elements that can jump within the genome. So based on the existence of intermediate RNA during the transposition process, so transposable elements can be mainly divided into two classes. Class 1, retrotransposon. It includes LTR retrotransposon line or sign. Then the second class is DNA transposon. It includes TIR transposon and helictron. So T are prevalent in eukaryotic genomes. For example, around 85% of the maize genome are occupied by TEs. So besides, TEs are key players in genome evolution and development. Many methods has been used for TE annotation in the genome. So before that, I want to give you a concept of what is TE consensus sequence. So because TE are repetitive elements, we can take all the copies from the genome and align them and generate multiple sequence alignments. And based on this alignment, we can generate a consensus sequence. There is several database collections have the TE consensus sequence from different species. We can take those replicates and run repeat mask on your genome. Then you will have TE annotation. So this method is called the repository-based method. But the limitation for this is you can only identify the TEs that exist in those database. And there's several de novo TE annotation methods. For example, self-comparator methods. You do self-alignment of the whole genome. For example, here this is a dot plot of the genome. By this way, you find the repetitive regions in the genome and then identify transport elements. Softwares like Grouper or Recon use this method. Or camera-based, for example, like Repeat Squad or pCloud, can also identify the transport elements in the genome. Another one will be the structure-based method for TE annotation. First, they identify the existence of LTR, the long-term repeat sequence, or the TIR, like LTR finder, harvester, retriever, or TIR learner and TIR finder. So beside that, there are also pipeline tools that combine different methods for TE annotation, like EDTA, repeat modular, repeats, air gray, or high TE. The output from them normally are better than the single strategy-based methods. So if you only do very simple TE masking for your genome, I think the output from those pipeline tools are sufficient. But if you want to do further analysis on TEs, for example, you want to analyze the TE divergency or TE evolution to perform evolutionary analysis on TEs, then the outputs from them, they are not accurate enough. For example, you could have a TE consensus sequence from those tools that cannot cover the entire TE sequence. If you want to perform analysis on this sequence, you have to get the intact version. In the traditional way, you can do many curation of transposable elements for it. So here I will give you a general introduction what manicuration transposable elements is. Because TE are repetitive elements, you have a query, and then you do BLASM search. You can find all the copies from the genome. Then you take all the copies out from the genome and do multiple sequence alignments. After you do extension for the left side and the right side of the multiple sequence alignments, until you find the potential boundaries for this TE. Then you check, find the boundaries here that can separate the highly and lowly conserved regions of the multiple sequence alignments. Of course, you can also check the target site duplication and check for the LTR retrotransposons. It can always start with TG and end with a CA, for example. Then you generate the condensed sequence. So this is the general steps for the manual curation of transposable elements. I highly recommend you to read those two papers, which can definitely help you to understand what manual curation of TE is, and this can also help you to understand the TEtrimmer. The manual curation of TEs can dramatically increase the quality of your T condenser library, but on the other hand, it's also very labor intensive and time consuming. So it's not possible to widely apply it on all the genomes that has been sequenced. For example, in the DFAM database, currently around 0.7% of the sequence are manually curated. I also did manual curation by myself before the development of TEtrimmer. I did it on a relatively small fungal genome, but I spent 3 months on that. Actually, this is the reason why I want to develop a software to automate this process. So here's the TEtrimmer workflow. So the input is the T condenser library. So for each sequence in the library, it will do BLAST search and find the copies and do multiple sequence alignments. Then it will cluster the multiple sequence alignments. Later I will show you why and how, and then come to extend the multiple sequence alignments. We said before, this step can find the potential boundary for the transport elements, and do multiple sequence alignment cleaning, and do a series of plotting function, and then TEtrimmer also provides a graphical user interface application to check and improve your results. So in this video, I will not cover all the steps for TEtrimmer. So I recommend you to read TEtrimmer paper. Definitely you will find more details there. So today I will only cover 4 main points here. The first is, how does TEtrimmer cluster sequencing the multiple sequence alignment? Of course, I will say why it's necessary. And the second is, how does TEtrimmer selectively extend the multiple sequence alignment sequence? And comparing with the traditional manual curation, why the selective extension of multiple sequence alignment is necessary. And then I will introduce you why we need to clean the multiple sequence alignment of TEs and how TEtrimmer will do that. Then, because TEtrimmer is designed to automate and replace the manual curation of TEs, and how could TEtrimmer make sure the output from it is accurate, is comparable to the manually curated libraries. Then come to the first one. How does TEtrimmer cluster sequence in the multiple sequence alignment? Here I give a reason why it's necessary. So in the traditional manual curation of TEs, here's a step. You have your query, do BLAST search, and do multiple sequence alignments. But in the real case, if you not have the multiple sequence alignments like this, which is very clean, but you will always have something like this. It is because the sequence similarity among the different T subfamilies, they are high, and after BLAST search, it will always take out the different T subfamilies out. You can see that after multiple sequence alignments, here you show you different alignment patterns. So you have to separate them before the extension of the multiple sequence alignments. Actually, this step is not easy. I tried different softwares, let's say CD Hits or Tree Cluster, but none of them can very accurately to do this task. To solve this problem, TEtrimmer introduced the algorithm to very accurately to do this. It combines the phylogenetic tree and with machine learning DBScan methods to cluster the multiple sequence alignments. So simply speaking, TEtrimmer will take out the divergent columns from the multiple sequence alignments and put it to IQTree, a software. It will build this phylogenetic tree. After, it will calculate the relative branch distance among each pairs of these multiple sequence alignments and generate a distance matrix. Then the distance matrix will be the input for DBScan, and then to cluster these multiple sequence alignments. Here you can see the results. It can very efficiently to do this task. So by default, TEtrimmer will only use the first 2 clusters for the further analysis. It will first sort the clusters according to the sequence number that is included in each cluster. It will only use the top 2 for example, further extension and boundary definition. You can modify the number by one option is called max class number. If you want top 3, then you can modify this one to 3, of course. Then this is the first setting point for TEtrimmer, how it do multiple sequence alignments clustering of T's. Then come to the second one, and how does TEtrimmer selectively to do sequence extension of multiple sequence alignments of T's. So in the traditional manual curation, you do multiple sequence alignments, and obviously you have to extend the left side and right side of the multiple sequence alignments until you find the boundary. But in the real case, you have to iteratively to extend the multiple sequence alignments. Let's say you did first round a sequence extension, but you find that extension is not sufficient. You cannot cover the boundary. Then you will do the second round sequence extension. So here I only show 2 rounds, but in the real case it can be 3 or 4 and 5 or 6. But there's several limitations for this extension strategy. So first extension, after every round of extension, you have to align all the sequence to check if this round extension is sufficient. Which means can cover the boundary of the transposable elements. Because manual curation, the multiple sequence alignments is very computational resource intensive. This can take a long time. Think about that. If you have to extend more than 5 rounds, this will take so long time. And the second limitation is, for example, in the first extension, because T are fragmented sequence, after first round extension, for some of the sequence probably will be not required for the second round sequence extension. So later I will show you how TEtrimmer to solve those 2 limitations. So for the first limitation, so TEtrimmer will not do the left side and the right side extension at the same time, but it will only do the left side extension first. For example, if you do first round extension, let's say extend 1000 base pairs, then it will not align all the sequence. All sequence means the initial sequence plus the extended sequence. Rather, it will only align the extended parts. So this can dramatically decrease alignment time and quickly give you if this extension is sufficient for this size. If this is not sufficient, it will do the one more extension. Same like before, it will only align this part and give you if the extension is sufficient or not. Then when you finish the left side extension, it will come to the right side. We'll do 1 round and the second round. Then by this way, it could quickly give you the extension number for left side of the multiple sequence alignment and for your right side extension, the multiple sequence alignment. Then this can decrease the multiple sequence alignment time and can save your time and save your computational resource. Then come to the second limitation. We say that, for example, for the top 2, here's the top 2 sequence. Let's say they are fragmented sequence. After first round's extension, for those 2 sequences that possible, only 1 round extension will be sufficient. So in the next round extension, you only need to extend the rest of the sequence, but not the top 2. So to solve this limitation, let's zoom the left part here. This is after the left extension after first round extension. We find for those sequence, the only extend 1 round will be sufficient because you can already see the alignment for them. They are lowly conserved. So it's not necessary to do further extension for those sequence, but you only need to do extension for those sequence. So for next round extension, you only do extension for those sequence and come to this result. You check it again, and for those sequence is same like before. You do not need to extend them again for the third round extension. Until here, you find the boundary. By this strategy, you can save your alignment resource, and this alignment strategy can also help you define the T boundary, especially for those highly degraded elements. Let's say 5 ends of line elements that can be highly degraded. By this extension strategy can help you to find the accurate, the 5' ends boundary for it. So this is the second selling point for TEtrimmer. Then come to third one, and why is it necessary to do multiple switch alignment cleaning for teeth, and how TEtrimmer do that. So after extension, and you will do multiple switch alignments again, and to define the boundary. But after that, you will always have the multiple switch alignment looks like this. That many gap columns are introduced, and many low conserved regions exist. So before the boundary definition, you have to clean them. So first is the gap column. You want to from here to here. To clean the gap columns, there's many softwares available. For example, let's say T-Coffee, CNLine, and so on and so on. The main idea is, for example, you have these multiple switch alignments and with lots of gap columns, you set a threshold. If the gap occupies more than 80% in this column will be eliminated. You will from the left side to right side to be here. But the problem you find that this region cannot be deleted. Someone will say, okay, probably you can decrease this threshold number. Let's say decrease this to 50%. If the gap occupies more than 50% of this column will be deleted. If from here to here. You see, the problem is those regions you want to keep them, but they are deleted. So it's not easy to set fixed threshold number to do this task in automated process. So in TEtrimmer, it introduces a new function or algorithm. It's called remove gap columns. It will first check, okay, if the gap occupies more than 80% in this column, definitely this column will be eliminated. Then it will check the columns that gap occupy 40% in this column, for example, in this column. And then it will check which one is the predominant nucleotide in this column. This is adenine. And then it will check if the predominant nucleotides occupy more than 70% of all the nucleotides. So if so, this column will be kept. Same for this column. This is guanine. And the guanine is predominant in this column, but it's occupy more than 70% of this column, and this column will be kept there. It will also check if the gap, if one column that has less than 5 nucleotides, and this column will also be eliminated. Then you can from this situation to here. You can modify those two parameters in two options of TEtrimmer. One is called gap thresholds, and one is called gap nucleotide thresholds. Then after gap column cleaning, you view from here to here. Then you have to clean the lowly conserved regions of these multiple switch alignments. You want from this situation to be here. So I tried to find software to handle this task, but I didn't find one. To solve this problem, TEtrimmer introduced also a new algorithm, two functions to clean the lowly conserved region, or we can also clean the rows of these multiple switch alignments. The first is called crop end by divergency. Let's say this is the starting region for this multiple switch alignments. You zoom it, come to here. So TEtrimmer, or the crop end by divergency function, will convert the entire multiple switch alignment to a proportion matrix. Let's say for the first column, in this column, the adenine occupy 40% in this column. All the adenine corresponding position will be upon 4. The gap will be 0. The thymine occupy 30% in this column. This will be upon 3, and the same for rest of the columns. There will be proportion matrix. And then for each row of this proportion matrix, this function will generate a sliding window. And it will move nucleotide by nucleotide or position by position and stop until the sum of the proportion number included in the sliding window is greater than the threshold. So there's two options you can set it in TEtrimmer. One is crop and divergence window. The default is 40. That means this window is 40. And the other one is crop and by divergence threshold. It's from 0 to 1. It's a proportion number. In this case, it's 0.7. So you use 40 multiply 0.7 is 28. That means when the sum of the proportion number that is included in this slide window is greater than 28, the slide window will stop. And all the positions from here to here, not there. So from here to here, that will be eliminated. So this is only one rule for the left side, and same for other rows. And for the other side, for the right side, the same. The slide window will be selected, created, and but move from right side from right to left. Then you can from here to here can very accurately, efficiently clean the multiple switch elements. And besides this function, there's another function. It's called crop and by gap. So this function will not convert the multiple switch element to a proportion matrix, but if you just take advantage of the original multiple switch elements, by first class you will see, okay, those regions, they are lowly conserved, you want to clean them, and in the traditional we have to select them and clean them. It's very, very time consuming. But when you have a look at that, you can, on those regions, there's always gaps exist there. But in the conserved regions, there's no gaps or very less gap. And for each line or each row of the multiple switch alignments, for example, in this one, same with the last function, it will create a sliding window, and it will move nucleotide by nucleotide for each line or each row, the multiple switch alignments. It will only stop until the gap number included in this sliding window is smaller than threshold. By default, the sliding window size is 250. You can modify it by this option. And the threshold is 0.1. That means this sliding window will stop until the gap number is smaller than 25. And all the nucleotide from here to here will be eliminated later. So this is the left side for the multiple switch alignment. And same for the right side, it will create another sliding window, but move from right side to left side. You can see from here to here can also efficiently to clean the multiple switch alignments. The magic thing is you can combine those two functions. You combine the crop and by divergence and combine the crop and by gap. This can give you a very, very clean result. Actually, this is the third selling point, and this is also the core selling point for TEtrimmer. The reason why we name it as TEtrimmer is because we introduced very efficient or innovatively introduced the multiple switch alignment cleaning functions or algorithm into this tool. And then it's come to the fourth point, the how to make sure the outputs of TEtrimmer are accurate. Let's say, because TEtrimmer want to automate the process, and how could we make the outputs from TEtrimmer is comparable to the manually curated results. The first method to solve is give you plots, reporting plots for each output. For example, for A and B, they are multiple switch alignment plots. In the first plot, it will zoom the starting region and end region of the multiple switch alignments, and also indicate where is the boundary that is defined by TEtrimmer. In this plot, you can also check the totally side duplication. And then in the second plot, it will give you entire multiple switch alignment plots, can tell you what the internal alignments looks like. And then TEtrimmer also introduced the T8 package. For example, here can give you, okay, before TEtrimmer analysis, this is the blast plot, and after indeed is the blast plot. And same for the dot plot. And then it's the before TEtrimmer, here's the OF and the PFAM domains, and this is after TEtrimmer, this OF and the PFAM domains. The final plot is called dot plots. the y-axis is the sequence before TEtrimmer analysis, and x-axis is after. So combine those plots. This can quickly give you information. For example, if the boundary definition decided by TEtrimmer is right or not, if you are satisfied with the results, and you can also quickly give information where and what is the difference between the sequence before and after TEtrimmer analysis. Just in case, based on the report plots, you are not satisfied with results. For example, the boundary definition is not right, and the extension has been done by TEtrimmer is not sufficient. TEtrimmer also provides a graphical user interface tool for this situation. So I will not cover a detail how to use this tool. I already provide another video. It's called the TEtrimmer GUI introduction. You can go there, and there I exactly showed you how to improve or check the TEtrimmer outputs. For example, why there's 4 files correspond with 1 t-consensus sequence. So combine the reporting plots and this TEtrimmer GUI. This can quickly give you information if you believe it. If you are not satisfied, how you improve it quickly. Then to come to the TEtrimmer workflow again. So the input is t-consensus library, and for each of the sequence in the t-consensus library, it will do BLASN search. Because TEtrimmer takes the advantage of the multiple sequence alignments, the repetitive property of the multiple sequence the TEs, they require at least 10 copies of the T exist in the genome. Otherwise, it will check if the terminal repeats exist and was numbered for length BLASN. It will decide if this is a low copy TE. Then, if you do multiple sequence alignments, I think now you already understand why we need to do multiple sequence alignments clustering of transposable elements. Then it also have to make sure that after multiple sequence alignment clustering, for each cluster there's more than 10 sequences included. Then after to enable to include the T boundary, you also need to do multiple sequence alignment extension. And now you are clear why we need to do multiple sequence alignment cleaning. And here this is generated plots, do ORF and PFAM domain plots, do T classification, output evaluation and deduplication. And for those steps, I will not cover by this video. So you can just go to our TEtrimmer paper and read it. Definitely you can find more details there. So the output, TEtrimmer will also give you a t-consensus library, which is much better than your input t-consensus library. You can definitely use it directly for your genome masking. But TEtrimmer also provides a GUI tool or application can help you quickly check and improve your results. So TEtrimmer, I would say, is not a simple pipeline, Python pipeline, but also includes lots of the core innovative algorithm inside. Can automatic the manual curation of transposable elements and give you a very high quality results. So here's this TEtrimmer GitHub is open source software, and it's free to use. Of course, you can run it on Windows. So here you need a WSL. You can run it on Mac OS and Linux. It's very easy to install it. So thanks to Han. So she packed the TEtrimmer to Conda already. You can install it by Momba and also run TEtrimmer. It's very easy. So you can use TEtrimmer input file and a genome file. Only need these 2 options here. So TEtrimmer also provide a continuous analysis function. If there's stop somewhere, you can just definitely start from there. You do not need to start to run it from the beginning part. So TEtrimmer also provides a singularity image and Docker. So sometimes, the conda package or the singularity image also include the source code of TEtrimmer, but maybe, the the version included inside that is out of date. That's not the brand new version. In this way, you can download or clone the current TEtrimmer version and run them inside the conda environment or the singularity image. For this, I already provided the detailed code in our TEtrimmer GitHub, so you can have a look there. Yeah, many thanks for your listening. I hope this can help you.